My name is Michaela Lee. I am a fourth-year microbiology major, and my thesis is on arsenic methylation of marine cyanobacteria, Prochlorococcus, and Synechococcus. I work under the mentorship of Dr. Stephen Giovannoni in the Department of Microbiology. For some background to this thesis, marine cyanobacteria in our oceans dominate global nutrient cycling. For this thesis specifically, we will be focusing on two organisms called Prochlorococcus and Synechococcus. Prochlorococcus and Synechococcus are some of the most abundant phytoplankton in our oceans. They thrive in oligotrophic waters, which means that these ocean waters are nutrient scarce. The total abundance of these two cyanobacteria are about 3 by 10 to the 27 and 7 by 10 to the 26 cells respectively. Marine cyanobacteria play a critical role in energy transfer from our primary producers to bacteria, and as I mentioned before, they are in oligotrophic waters where nutrients are harder to come by. Critical nutrients such as phosphate are limited, and because these critical nutrients are so limited, they can create high ratios of toxic elements such as arsenate. Arsenate and phosphate are chemical analogs. Chemical analogs mean that between two compounds, there is only one thing that makes them different. So in the pictures on the right side of the screen, we have phosphate, which is the top picture, and arsenate, which is the bottom picture. The only difference between these two compounds is their center atom. When we have high ratios of arsenate to phosphate, sometimes we see accidental uptake of arsenate. Arsenate can compete with phosphate in energy metabolism in our cyanobacteria. Some effects of arsenate competition with phosphate inside of a cell include the decoupling of oxidative phosphorylation, and that interferes with ATP production. As I said before, we are working with two model organisms, Prochlorococcus and Synechococcus. Prochlorococcus is a unicellular marine cyanobacteria. For this experiment, we used one low-light strain, 9313, and one high-light strain, Med4. These are axenic, which means that it's just that one strain in a culture. These are non-motile cyanobacteria, and you can find these around the subsurface layer of oligotrophic gyres. They're considered a finicky bacteria to culture, and they utilize divinyl chlorophyll A and B as light harvesting complexes. We are also focusing on Synechococcus, which is a unicellular marine cyanobacteria. I will be referring to it as WH8102. This strain is motile, and the motility is a response to nitrogenous compounds, not a light gradient. Synechococcus also does utilize light harvesting complexes such as chlorophyll A. And here are just a couple of pictures of what these two cyanobacteria look like. When we think about high ratios of toxic elements to critical nutrients, and how that relates to energy transfer between primary producers and bacteria, there is a specific parasitic arsenic cycle between heterotrophic bacteria called SAR11 and our marine cyanobacteria Prochlorococcus. A simple overview of this pathway is that arsenate, which is in our waters, in our environment, is taken up into the Prochlorococcus cell through phosphate transporter proteins. This arsenate, or ASV, is reduced to arsenite, or AS3. Arsenite can then be further methylated into monomethyl arsenic acid, MMA, and dimethyl arsenic acid, DMA. Then we see the excretion of these methylated products back out into the environment. The heterotrophic bacteria SAR11 cell picks up these methylated products and through some unknown processes is able to demethylate these methylated arsenic compounds and release back out arsenate into the environment. This is dubbed a parasitic arsenic cycle, as we hypothesize that between a phytoplankton cell and a SAR11 cell, there is some type of parasitic energy gain for the SAR11 cell. The objectives of this study were: how do different ratios of arsenate and phosphate impact cellular growth, and how do cyanobacteria tolerate arsenate? We hypothesized that as the ratio of arsenate to phosphate increased in laboratory conditions, the cellular growth rate and density would decrease. This would make sense as arsenate is a known toxic element, especially to cyanobacteria. We also hypothesized that cyanobacteria do utilize a variety of pathways, such as detoxification, methylation, and efflux pathways, to handle arsenate when discriminating between arsenate and phosphate if there's accidental uptake. The methodology of the study includes some very specific growth conditions. As I mentioned before, these cyanobacteria are finicky to culture. Growth conditions include a 12-hour light and dark cycle for our highlight strains, MED4 and WH8102. They are grown at about 50 to 55 microeinsteins. For our low light strain, 9313, it's grown at about 20 microeinstein. All cultures were grown in 15 milliliters of media, but they were housed inside of 125 milliliter loosely capped flasks for gas exchange. All culture flasks were held at about 18 to 20 degrees Celsius. Quadruplicates for 6 different treatments of ratios of arsenate to phosphate were created. The ratios included 0 times arsenate, 0.2 times arsenate, 1 times arsenate, 5 times arsenate, 10 times arsenate, and 50 times arsenate. These ratios were calculated using a 0.01 molar arsenate stock and a 0.01 molar phosphate stock. The concentrations of arsenate for Synechococcus ranged from 0 millimolar to 1.18 millimolar. For Prochlorococcus, the arsenate concentrations ranged from 0 millimolar to 2.5 millimolar. The results of this experiment were to compare the growth curves of these 3 strains of bacteria. This first graph shows Synechococcus WH8102 growth curves, and as we can see in this graph, it appears that between all 6 of our growth treatments, that there wasn't much difference in growth rate or cellular density. Pretty much all of our treatments reached low 10 to the 8th cells per milliliter, and this experiment ran over about 18 days. We also saw similar results for our Prochlorococcus strains. This is MED4, our highlight strain. Over 2 months, we saw pretty similar growth rates and final cell densities. However, MED4 was able to reach a higher cell density, around 5 by 10 to the 8th. Some culture flasks did reach up to around 1 by 10 to the 9th cells per milliliter. Then finally, we have our low light strain 9313, and this is the most interesting graph. As we can see, for the 2500 micromolar arsenate concentration, it appears that these treatments could not get past 1 by 10 to the 6 cells per milliliter. This indicates that there could be some sort of threshold tolerance for a low light strain specifically. For this study, we wanted to, 1, confirm that there are capabilities for cyanobacteria to handle arsenate, and 2, to hypothesize the types of pathways that cyanobacteria can use to tolerate arsenate. So here we have a table of genes that correlate to arsenate tolerance and how they are present in different strains of cyanobacteria. So going down the table, we have the gene PSTS, which is phosphate uptake. We see that this gene is present in both Synechococcus and Prochlorococcus, We would hope that that is present, as phosphate is a critical nutrient for these cyanobacteria. Next, we have ARSC, which is an arsenate reductase. This reduces arsenate into arsenite. Again, we see a copy of this gene in all of these strains. Next, we have ACR3, which is an efflux pump for arsenic species. We see this in Synechococcus and our highlight Prochlorococcus med4. However, we don't see this in our low-light strain 9313. Next, we have ARSM, which is a methyltransferase. This is seen in all of our strains. And finally, we see ARSR, or an arsenic suppressor. This is an operon, which allows these cells to activate arsenic-related pathways if there is arsenic presence. And we also see this in all of our strains. The most important thing to note here is that our low-light strain 9313 does not contain an efflux pump. This means that if 9313 is taking up arsenate and is unable to get these toxic species out of the cell, then there must be some sort of threshold in which we saw in the previous slide. For future directions for this project, we want to take a look at how low phosphate cultures can impact our arsenate tolerance, and how do generational cultures handle arsenate tolerance. For this experiment specifically, we were only able to do experiments involving high phosphate cultures. Due to time constraints in running these experiments, we stuck with the high phosphate culture. However, if we were to lower our phosphate concentrations in cultures, we are wondering how these cells might be changing these pathways in order to better tolerate arsenate over longer periods of time. We also want to find out what kinds of arsenic species are being produced by each strain. We originally were going to do this, but again, due to time constraints, we were unable to do that. And this would help confirm how each Cyanobacteria species was able to handle these arsenate and what kinds of species were being produced. And then finally, we want to do a genomic comparison of our laboratory strains to in situ, which is just from the environment or eutrophic waters where we find these cyanobacteria, and how many copies of these arsenic-related genes do we find in laboratory strains. For acknowledgements, I would first like to thank my wonderful laboratory group in the Giovannoni lab. Your support and advice has been invaluable, and I appreciate all of you. Thank you to my postdoctoral mentors, Dr. Chi-Ping Lee and Dr. Holger Buchholz, for training me and giving a solid foundation in marine culturing for this project. Thank you to Dr. Kimberly Halsey and Dr. Philip McFadden for agreeing to be on my thesis committee. And lastly, but not least, a very special thank you to my PI, Dr. Stephen Giovannoni, for taking a chance on me and providing wonderful guidance on this project and an incredible laboratory experience.